Hello students, very good afternoon. So welcome to today's session. Before we start the session, I would like to have a technical check on the video and audio. Please let me know if the audio and video is clear so that we can continue on with today's session. You can even put your comments on the chat box. So if there is any technical issues, we can correct. All right, students. So let's carry on with today's session. So if you remember the last class, we were talking about mechanism of urine formation. And in mechanism of urine formation, we came across some of the important topics where we studied three important phases of urine formation. That is through glomerular filtration, reabsorption, and tubular secretion, right? So as I said, in glomerular filtration happens in the renal corpuscle. So the afferent arteriole, which is carrying the blood into the glomerulus, and we know that glomerulus is a tuft of capillaries. So as the blood passes into this glomerulus, now there is a lot of blood pressure within this, capillaries. So the difference in the blood pressure in the lumen of the Bowman's capsule and the tubules of the glomerulus will create a pressure difference that will lead to the exchange of certain ions or filtrates. Okay. So usually whatever the filtrate that is formed is usually the components of the plasma of the blood, except for the larger proteins. So that means there are no cells that is coming out of this glomerular tubules. Correct? No. So when we talk about what is the filtration process and who facilitates it, so we understood that the layers of your glomerulus and the Bowman's capsule make one integral membrane, okay, that will create as a filtration, okay, filtration slit. So here we have three important membranes. One is endothelium, second one is epithelium, third one is basement membrane. So endothelium of glomerular blood vessels, epithelium of Bowman's capsule, and basement membrane between these two layers. You can see clearly here in this given picture. So we have endothelium here, correct? No? And we have basement membrane here, and then we have epithelium here. So these three layers combined together, they form filtration of blood. Okay. So now, when we look at one of the important aspects of this arrangement of this membrane, so if you look at epithelial cells of Bowman's capsule, okay, the cells of epithelia of Bowman's capsule are called as podocytes, and they're arranged in such a manner that they will leave some of the minute spaces between them. And these minute spaces are called as filtration slits or filtration pores or slit pores. Now, what is the significance of this filtration slits or slit pores? Now, the pore size is sufficient enough to allow all the particles of plasma except for larger proteins. Okay, so that is why this pore size of this filtration slits are very, very significant in undergoing the filtration process. Okay, so as I said, if you see what are the components that enters the lumen of the Bowman's capsule after it passes through the capillaries of the glomerulus. See, this is, let us assume that this is the afferent arteriole and this is the efferent arteriole. So as the blood moves here, right? So there is a filtrate that is accumulating in the lumen of the Bowman's capsule. Now, what is accumulated at the lumen's capsule? Every constituent of the blood plasma except the larger proteins. Okay, so that will pass through the lumen of the Bowman's capsule. Now, whatever is accumulated in the Bowman's capsule, it is called as filtrate. So if you see, our kidneys will generate around 1,100 to 1,200 ml of blood, okay, is filtered for every minute. That means 1/5 of our blood that is pumped out by our ventricles, okay, are filtered by our kidneys. Correct? No. So now, if I take 1 minute, and if I see what is the amount of filtrate that is formed here in the lumen of the Bowman's capsule in 1 minute. So that is the definition of glomerular filtration rate. What is GFR? It is the amount of glomerular filtrate that is formed per minute. It is called as glomerular filtration rate. Now, this glomerular filtration rate is very, very essential to understand the normal physiology and functioning of our kidneys. So for a normal person, the normal glomerular filtration rate is around 125 ml per minute or 180 liters per day. Okay. So this is about glomerular filtration. But if you see, out of 180 liters of the blood that is filtered, right? So are we eliminating 180 liters of waste every day? Is it possible for our blood, for our body to accumulate so much of fluid? It's not possible practically. Okay. So even if you see the entire volume, like reduce it, okay, to one filtration. So that means you will eliminate all the fluids in your body. So that is why what happens, out of this 180 liters of the blood that is filtered, it has to undergo series of reabsorption. Okay. Reabsorption is where all the essential things are taken back into your blood circulatory system, or the non-essential things are concentrated and it is concentrated as urine. So if you see, we are filtering around 100% of this 180 liters, we produce around 99% of this filtrate that is produced is reabsorbed. Okay, and it is recirculated back into our body. Okay, so only around 1.5 to 2 liters of urine is eliminated in our body on a daily basis. Okay, so this reabsorption is a very important process. Why? So from the filtrate, some of the important components like glucose, amino acids, sodium ions are reabsorbed, okay, actively. Whereas when you look at the nitrogenous waste are absorbed passively. That is where reabsorption is very important. But if you see for our body, okay, the fluid balance happens with the availability of water. So the water levels in our body are very, very important for many aspects. Okay, so it is a primal factor for osmolarity in our body. So that is why if you see, the water absorbs passively in the initial segments of our nephrons, and in the later phases of the nephrons, it goes through active means of absorption. So let us see where the reabsorption process takes place. Once the filtrate that is accumulated in the lumen of the Bowman's capsule, we know that the lumen of the Bowman's capsule will lead into the tubular structures called as the first initial segments of this tubular network is our proximal convoluted tubule, correct? No? So if you see what is the function and the contribution of proximal convoluted tubule, see proximal convoluted tubule, it absorbs most of the nutrients, up to 70 to 80% of electrolytes and water. And this is possible for the proximal convoluted tubule to do so is because it is lined by a specialized cells called as simple cuboidal, okay, ciliated epithelial cells, or that is also called as brush border epithelial cells. So that will increase the surface area of the PCT for reabsorption. Okay. Whereas if you look at what does loop of Henle do, we know that the loop of Henle is responsible. See, we have loop of Henle, we have the descending loop of Henle, and we have the ascending loop of Henle. Okay. Each of the loops, they have a specific function to perform. And that depends because of the way the cells and the way the tubes are organized. If you look at the descending loop, you can see that the initial segments of the descending loop are made up of thick segments followed by the thin segments, right? The tube is thin. Whereas if you see the ascending loop, the initial segments is made up of short, thin segments and long, thick segments. So what does this define? The thick and the thin segments define the permeability of water. So the thin segments, they are highly permeable to water, whereas the thick segments are impermeable to water or less permeable to water. So if you look at the loop of Henle, right, as I said, it is very important in maintaining the osmolarity, okay, and medullary interstitial fluid volume. Okay, so let us see what does the descending loop do. So the descending loop, okay, is permeable to water, whereas it is impermeable to electrolytes. So what happens whenever the water is removed out of your descending loop, the amount of solvent in a solution decreases. When the solvent concentration decreases, the solute concentration increases. That means the filtrate is concentrated as it moves down the loop. Whereas when you look at the ascending loop, here the maximum absorption takes place and it is impermeable for the water to move across. So here only the ions, electrolytes are transported. So here what happens, since the electrolyte exchange will happen, that is why the filtrate gets diluted in the ascending limb. So the last portions of your nephron, that is the distal convoluted tubule and the collecting ducts. So they also have a significant role to play. If you see the DCT is very conditional for its reabsorption based on the requirements and based on the overall body condition. Now the DCT functions in the reabsorption of sodium and water in uptake. Okay. Whereas when you look at the collecting ducts, we know that the collecting ducts are interconnections between not one nephron, it is several nephrons are interconnected for the collecting ducts. So here collecting ducts, they extend from cortex into the medullary regions of the kidney. Okay, and finally they go and collectively they connect to your renal pelvis, and then it is connected to ureters. Okay, so here what does collecting ducts do when they are near the medullary and the cortical regions? Here, whenever our body requires, for example, when you are dehydrating a lot, so that is when the maximum reabsorption of water takes place. Okay, and the concentrating of urine takes place near the collecting ducts. Okay, so it also allows the passage of urea, okay, into the medullary interstitium so that the osmolarity is balanced. Now we understood what is glomerular filtration and reabsorption. So another important aspect for concentrating the urine and for urine formation, okay, urine formation takes place. Urine is not formed the moment they enter the kidneys. Okay, it's a process. Okay, it's a gradual process. Here, what is happening? The final process of urine formation is tubular secretion. Now, what is tubular secretion? Usually, the cells of proximal convoluted tubule, distal convoluted tubule, and even your collecting ducts, they secrete some of the ions, okay, which help in maintaining the osmolarity, pH, and acid-base balance. Okay, so let us see what they do. If you see PCT and DCT, they maintain ionic balance, that is sodium and potassium balance they maintain, okay, by secretion of sodium or potassium ions based on the requirements. And they also maintain acid-base balance. Okay, they also maintain acid-base balance, that is a pH balance. How do they maintain this pH balance? Either by secretion of H+, K+, or NH3 ions into the filtrate, and they also support the reabsorption of bicarbonate from it. But if you see the collecting ducts also play a major role in maintaining pH and ionic blood balance, ionic balance in the blood. Okay, how do they do it? By secretion of H+ and K+ ions. So this secretion is called as tubular secretion. So let us go for the next topic, that is urine formation and countercurrent urine formation, that is also called as countercurrent multiplier system. Okay. So let us see what happens. What is countercurrent multiplier system? If you see how our nephrons are organized, after the exit of your, okay, after the exit of the PCT, see that once the PCT comes, it leads to what? Loop of Henle, isn't it? See the arrangement of loop of Henle, it first descends and then it reascends, correct? No? First, initially they have thick segments followed by thin segments. Here they have thin segments followed by larger thick segments. So this is one arrangement. And the second arrangement is, look at how the vasa recta is organized. The vasa recta surrounds the loop of Henle in the similar fashion. Okay. But if you see the movement of substances across these two structures, it is quite in an opposite direction. So let us understand very clearly. So what is countercurrent mechanism? Here the concentration of urine happens because of Henle, loop of Henle and vasa recta. Okay. Now what happens? The filtrate that flows through the two limbs of Henle's loop, okay, and the blood that flows through the two limbs of vasa recta are in opposite direction, as I said. So if the direction is here like this in the loop of Henle, and this will be in the opposite direction in the vasa recta. Okay, so that is why we call it as countercurrent multiplier system. So here the movement of substances are also in the opposite direction. Okay, fine. Let's see how the countercurrent mechanism actually works, right? So what are the important criteria for the counter current mechanism to even happen. First one is the proximity. Proximity means the distance between the loop of Henle and the vasa recta. So this proximity, it will lead to the change in osmolarity. What type of osmolarity? If you compare the medullary region of the kidney and then you have the cortical region of the kidney, as you move from cortical region to medullary region, the osmolarity tends to increase. You can see here from 300 to it is increasing to 1000, 1200, correct? No. So this increase in osmolarity takes place because of the proximal arrangement of this vasa recta as well as your loop of Henle. Now, in the cortex, the osmotic pressure that is around 300 milli osmo per liter, it increases gradually as it goes through the medullary interstitium to 1200. So the change in this pressure gradient will create is caused because of the sodium chloride as well as the urea that is exchanged between these limbs. Okay, so let us see what happens in the first stage. First, we have to talk about NaCl. So NaCl in the ascending limb, you can concentrate here. NaCl in the ascending limb of the loop of Henle is exchanged. See, the loop of Henle in the ascending limb, they give back NaCl to the interstitium. Okay, so as the NaCl accumulates in the interstitium, what is the interstitium? Interstitium is the space that is between the two loops, right? So the space that is occupied by the portions where the nephrons are not, the gaps in the nephron are called as interstitial space. Okay, so as the NaCl is exchanged by the ascending loop, okay, now what happens? It will accumulate in the interstitium. Okay, now similarly, if you look at the urea, okay, from the thin segments of the ascending loop, okay, see, if you see urea, right, where is urea? Yeah, we have urea. So the urea enter the thin segment of the ascending loop. Okay, again, after it enters the thin segments of ascending loop, it is transported back to the collecting ducts. Okay, it is transported back to the collecting ducts. Here, now what happens? Because the electrolytes like NaCl and the urea, okay, are retained in the interstitium. Okay, so that will help in maintenance of concentration gradient in the interstitial space. Okay, in this space, what happens? There is a maintenance of concentration gradients. Okay, so usually in the medullary interstitium, you can see that the gradient is keep on increasing. Okay, so as the concentration gradient increases in the medullary region, now what does it facilitate? It facilitates in the easy passage of water from collecting duct. Okay, see here from collecting duct, you can see that there is an easy passage of water. Okay, so where does it go? So as the water comes out from the collecting ducts, it is helpful in concentrating the urine as the urine goes from the cortical region to the medullary region through the collecting ducts. Okay, next one. Next, what is happening? If you see the overall aspect, the DCT and the collecting ducts, okay, they, because of this mechanism of exchange of NaCl and exchange of urea in the interstitium, and the change in concentration gradient will help in the movement of water, okay, and thus concentrating the urine. So here the two most important portions of the kidney, that is the DCT and the collecting ducts. Here, because of this mechanism, here the urine gets, or the filtrate gets more and more concentrated. How many times does it get concentrated? Around 4 times it gets concentrated. Okay, so initially the filtrate formed is only having 300 osmoliter, and then it changes to 1200 milli osmoliter. Okay, so this is how the exchange happens between the ascending loop, okay, and then it happens between the vasa recta, okay, and it helps in the concentration. See, whatever the water that is being released into the interstitium, it will be reabsorbed. Okay, it will be reabsorbed by your vasa recta and it will be taken back into the circulation. Okay, so here what happens, because of the ionic change, usually the urine is concentrated, okay, within the collecting ducts, and it is transported as urine. So now we all know that how the urine formation takes place. And the next important aspect that we have to understand is what happens when we have to release this urine. So the process of release of urine is called as micturition. Okay, by definition, micturition defines the elimination of urine from the urinary bladder, okay, through urethra. It is called as micturition. So here, what are the steps in micturition? First, what happens? Usually, our urinary bladder, okay, are made up of a very special type of muscles. Okay, so these muscles of the urinary bladder, they are filled with a special type of receptors called as stretch receptors. As the urinary bladder gets keep on filling, okay, as the urine in the urinary bladder keeps filling, now the urinary bladder becomes bigger and bigger. As it becomes bigger and bigger, that means the muscles of the urinary bladder are stretching. So that will trigger the switching on of the stretch receptors. Now, once the stretch receptors are on, now it will start an impulse. Okay, it will stimulate an impulse of this filling of urine in the urinary bladder, and it is carried out to the central nervous system. Okay, usually in our spinal cord, okay, there are micturition centers. Okay, they usually they have two of the centers, like sympathetic and parasympathetic centers, where they usually send a signal, okay, to your urinary bladder back to whether the muscles have to relax or they have to remain constricted. Okay, so let us see what happens. Okay, 1 second. Yeah. So what is happening once the stretch receptors increases, right? The stretch receptors will send a signal to the central nervous system. Where is the central nervous system? Central nervous system is your spinal cord, isn't it? So usually, okay, once it reaches the spinal cord, now what happens? Now your spinal cord will send a signal back for the constriction of the smooth muscles of the bladder, as well as relaxation of the urethral sphincter. Once the relaxation of urethral sphincter takes place, okay, then the urine will be released. So usually we have two urethral sphincters. One is internal urethral sphincter, and then we have external urethral sphincter. Understood? Fine. So usually in the internal urethral sphincter, it is involuntary, whereas external urethral sphincter, it is voluntary. That is when you can postpone whether you need to micturate or not. Okay, fine. So as we micturate, because of the causing of the reflex called as micturition reflex, so that will lead to elimination of urine. So around 1.1 to 1.5 liters of urine, okay, is excreted by an average human per day. So we know that urine should be transparent in color, but sometimes because our urine color may change, it may change to light yellowish to more yellowish color. As the yellow color changes, okay, it becomes more yellowish. That means the characteristics of urine has some problem. Okay, see, for example, it is light yellow, that means chloride ions are there. More yellow, sodium and potassium ions are there. Even more yellow, that means creatinine is eliminated, and even bicarbonates are eliminated. Understood? So the properties and coloration and the pH of the urine will determine the physiological health of a human being. Okay, so that is why we perform urine analysis. You can take an example of diabetes, right? So what do we test in diabetes? Glucoseuria. So the presence of glucose is determined, okay, by ketonuria bodies that is present in your urine. So that will indicate if a person is having diabetes or not. Okay, so there are other organs of excretion too, not just your kidneys. There are other organs such as lungs, liver, skin, etc. So let's see how they help us in the process of excretion. When it comes to lung usually helps in removal of carbon dioxide, right, by expiration. So usually around 200 ml of carbon dioxide is eliminated by our lungs, and even the water in the form of vapor is eliminated by our lungs. Next, our liver is called as a master gland because it metabolizes and it detoxifies so many biomolecules and biological compounds. Okay, some of the important functions of liver are we know that it secretes bile, which contains bilirubin and biliverdin, okay, cholesterol, and it is degraded into degraded steroid hormones, vitamins, and drugs are all processed in the liver. So usually most of them are passed to the digestive waste. So the brown coloration of the fecal matter is because of biliverdin. Okay. Next one is the skin. We know that the skin, usually we sweat. Okay. Through the sweat, we eliminate water, sodium chloride, and small amounts of urea and lactic acid. So usually whenever we are in a very high temperature regions, okay, that is when we start to cool out, okay, or eliminate out all the okay, heat in our body through sweating. Okay, that leads to cooling effect in our body. Whereas our skin also is embedded with some of the glands called as sebaceous glands or oil secreting glands. So they secrete sterols, hydrocarbons, and waxes in the form of sebum. Okay, so this sebum, the oily coating on your skin or your face, okay, so that is because of the sebum. So the sebum usually gives a oily covering to your skin, so which helps in protection against some of the common infections. And finally, saliva that is produced in our mouth, it has lysosomic activities. Okay, along with that, it also helps in elimination of nitrogenous waste. Okay, so next we have to understand another final aspect, that is what is the process of regulation of kidney function. So usually the regulation, that is how much of urine has to be formed, or whether the urine or the filtration process has to be fastened or it has to be slowed down. So this happens because of the hormone feedback mechanisms that is involved in our body. So usually some of the important aspects of our physiology that is involved in hormone feedback mechanism are: one is hypothalamus, and other one is juxtaglomerular apparatus that is near the nephrons, and next one is heart. So how does it react? Like what makes them to react? Usually the change in the body volume, okay, body fluid, blood volume, body fluid volume, or ionic concentrations. These are some of the factors that will trigger some of the receptors called as osmoreceptors in the body. So whenever these osmoreceptors are triggered, okay, it leads to regulation of this hormonal regulation of urine formation. So the first one we are trying to study is regulation of urine by ADH. ADH is also called as antidiuretic hormone, or it is commonly called as vasopressin. Okay, so here is what happens when you are dehydrating. Okay, when you're dehydrating, so now what happens? That means there is very less amount of water in your blood. So now the osmoreceptors will pick up this decrease in water volumes in your blood. Okay, so now what happens? The osmoreceptors in the hypothalamus, they signal the posterior pituitary. The posterior pituitary, okay, will stimulate and it secretes ADH, that is antidiuretic hormone, is secreted by the pituitary glands. Once they secrete antidiuretic hormone, now they make the kidney to retain more water. Okay, and how do they do this? By more absorption. Okay, more absorption of water will take place in the DCT and collecting ducts. Okay, that is one thing. So this process of reabsorption of water and concentrating urine is called as diuresis. Okay, so this increases the fluid volume. An amazing factor is these osmoreceptors that are activated in the hypothalamus, they're also responsible for stimulation of thirst. Okay, and that leads to drinking of water. Okay, that whenever you feel thirsty, that is because of your ADH and your hypothalamus that is helping you to understand this. Okay, now in case your fluid value or fluid volume is back to normality, now what happens? Osmoreceptors in the hypothalamus get switched off. So that will stop the signal from hypothalamus to posterior pituitary, and that will negate, okay, the production of ADH, and the ADH will not be released, and that will complete the feedback mechanism. So once the ADH usually reduces, okay, that will constrict the blood vessels, resulting in increase in BP and glomerular blood flow. Okay, so that is what happens. And the second one is regulation of urine formation through JGA, that is juxtaglomerular apparatus. So juxtaglomerular apparatus is a special body, okay, which is formed by the modification of cells of distal convoluted tubule. You can see here. Okay, we have distal these are the distal convoluted tubule, and this is the afferent arteriole. Okay, the cells of the afferent arteriole and the distal convoluted tubule, they are modified to form this specialized body called as juxtaglomerular apparatus. Now, the juxtaglomerular apparatus is in contact with the afferent arteriole as well as your distal convoluted tubule. That means the beginning portions of your nephron as well as your end portions of your nephron. Understood? So now what happens whenever the juxtaglomerular apparatus realizes that there is very less fluid volume. Okay, that means there is very less flow in the glomerular apparatus, blood flow in the glomerular apparatus. Okay, now what they do is they regulate the glomerular filtration by producing one of the factors called as renin. Okay, so this release of renin will stimulate a pathway called as RAS pathway, or it is also called as renin-angiotensinogen pathway. Okay, renin angiotensin aldosterone pathway. So let's see what happens. So this renin angiotensin mechanism starts like this. Whenever there is a fall in glomerular blood flow, right? So the glomerular activates the juxtaglomerular cells to release renin. So once the renin is produced, renin will convert the inactive angiotensinogen in the blood to active angiotensin 1. Okay. The active angiotensin 1 in the liver, in the lungs, it is converted to angiotensin 2. So the angiotensin 2 is a vasoconstrictor. That means it will make your blood vessels smaller. So as the angiotensin 2 is released, which is a vasoconstrictor, what does it affect? It affects the glomerular blood pressure. How? It increases the glomerular blood pressure. As you decrease the size of the blood vessels, the pressure of the blood will increase automatically, right? And thereby glomerular filtration rate also increases. And angiotensin 2 also activate adrenal cortex to release one more factor called as aldosterone. Now, what does aldosterone stimulate? Aldosterone that is released by your adrenal cortex, okay, it will lead to reabsorption of sodium and water, okay, from the distal parts of your tubule, that is distal convoluted tubule and the collecting ducts. So thereby it will increase the blood pressure and body fluid volume and even the glomerular filtration rate. Okay, and the final mechanism is by ANF pathway. What is ANF pathway? Atrial natriuretic factor. Okay, atrial natriuretic factor. What is this atrial natriuretic factor? So this is a factor that helps to check the renin-angiotensin mechanism or RAS pathway. Now, what is happening? Because of the increased blood pressure, right? Now, as the blood pressure is sensed in the atria of the heart, right? As the atria senses that there is more flow of blood in the heart, now that will make them release a factor called as atrial natriuretic factor or ANF factor. Now, once this ANF factor is released, it acts on the blood vessels and it will dilate them. Okay, it's a vasodilator. Okay, what is vasodilation means? Blood vessels will become bigger. As it dilates the blood vessels, the blood flow will get slower, thereby reducing the blood pressure. As the blood pressure decreases, now what happens? In turn, the glomerular filtration also drops. Okay, and then your body fluid will get eliminated at a faster rate. So this is a mechanism of urine formation, students. Thank you very much. We'll conclude this session. If you have any doubts, you can post your doubts in the chat box. I'll wait here for a few more minutes. All right, students, I will end the session now.